here for attending today's webinar, wherein we are remembering Har Gobind Khorana, the Nobel Laureate, on his birthday for his contributions for the betterment of mankind. And on this occasion, we have with us our chief guest of the day, Dr. S.S. Marwaha, Chairman, Punjab Pollution Control Board. Very warm welcome to you, sir, for attending today's webinar and for accepting our invitation. Dr. Nilima Jarrett, Director General, Science City. Our resource persons of the day, Dr. Felix Bust, Associate Professor, Central University of Punjab, Bathinda, and Dr. Vandana Jaiswal, Scientist, CSIR, IHBT, Palampur. So once again, I welcome our chief guest and our resource persons. And at the same time, I request Dr. Jarrett, Director General, Science City, to welcome our chief guest and to give introductory remarks. Ma'am, please.
Thank you, Kamal. Thank you so much. A very good afternoon, Dr. Marwaha, Dr. Felix, Dr. Vandana, all teachers, dear students, all the officers from various departments who have joined us. A very good afternoon and a very warm welcome to all of you. For us, it is a proud day that we are celebrating the birth centenary of one of our, in fact, I would say the only Indian Punjabi who has won the Nobel Prize. Punjab has always been very proud of its diaspora, which has brought laurels to the country. But one of the topmost luminaries, I would say, has been Dr. Har Gobind Khorana. He is not only the first Punjabi to win Nobel Prize, he is also the only Indian Punjabi. Although at the time when he won the Nobel Prize, he was already a US citizen at that time. Dr. Khorana, as many of you would know, shared his Nobel Prize with Dr. Nirenberg and Robert Hawley. They were all working in the University of Wisconsin at Madison, and their research was their research primarily showed the order of nucleotides in nucleic acid, which carry the genetic code. These days, we talk about DNA sequencing, genetic code, and things like that. And we have started using it in our day-to-day lives also. But at the time in 1968, when Dr. Khorana had undertaken this work with his colleagues, it was one of the most pioneering works, which has actually had a very big impact on human development and human biology. And on the same year, he was also awarded the Lucia Gross-Horwitz Prize from the Columbia University. And Dr. Khorana, as some of you might know, was born in Multan at that time, and that was pre-independence, so it was the combined Punjab. And he did his master's from the University of Lahore. And from there, he moved on to the US, from where he did all his pioneering work. So it is a great honor for all of us that we are celebrating his birthday today at the Punjab Pushpa Gujral Science City, and we are trying to take his legacy forward. I hope many of the children who attend today's program would be motivated to work on some of the areas on which Dr. Khorana had worked. I also would like to say that we are greatly honored that for today's program, our chief guest is Dr. S.S. Marwaha. He is himself a very famous biotechnologist. He is a PhD in microbiology, and his entire work is on industrial biotechnology and on fermentation technology. He has been the director of biotechnology in the government of Punjab from, I think, 1996 on to say about 2006 or so, if I'm correct, Dr. Marwaha.
Yes, yes.
it has been, I have been associated with him since a very long time, and it has been a great pleasure and also a very big learning experience to have worked with him. Uh, one, um, one big thing which we need to learn from Dr. Marwaha is primarily how to raise funds for any program which we want to do. And he has been instrumental in conceptualizing and in setting up what you now see as the knowledge city in Mohali. He was the one who conceptualized that the state should have all these institutions which do cutting edge research so that the state can be brought up on the research map of the country. He was also the first CEO of the Punjab Biotechnology Incubator. Uh, and he has also been at the Central University of Punjab. He has been the professor and head department of applied agriculture at the Central University Bathinda. And presently, Dr. Marwaha is the chairman of the Punjab Pollution Control Board, where again he has done a lot of pioneering work, especially in testing of water quality and improvement of water quality in the state of Punjab. We all know how important it is for all of us. And also a lot of pioneering work in the area of paddy straw management. It is indeed a great honor and a pleasure to welcome you, Dr. Marwaha. Thank you very much for your time. I know you are today also, you are in office, you are still working, but I'm really, really grateful, me and the entire staff of Pushpa Gujral Science City, for accepting to be with us here today and to talk to our students, to talk to the children, to talk to the teachers. Thank you very much and a very warm welcome to you. I also would like to extend a very warm welcome to Dr. Felix Bust, who is Associate Professor and Head of the Department of Botany at Central University, Bathinda, and Dr. Vandana Jaiswal. Dr. Jaiswal is a CSIR Young Scientist Awardee, and she's from the Institute of Himalayan Bioresearch Technology, Palanpur. I heartily welcome you, Dr. Felix Bust and Dr. Vandana Jaiswal. Thank you for agreeing to talk to our students. And last but not the least, I welcome all the students, all teachers, all academicians which have joined with us today, officers from several departments who have joined with us. And I hope that we would have many interesting things to learn today. And I'm sure that all of you would enjoy today's session. Without coming in between you and the chief guest and the speakers, I hand over back to my colleague, Dr. Kamal.
Thank you, ma'am. And thanks a lot, sir, for accepting our invitation. As I've already said, we are really happy to have you here. And Dr. Rajesh Grover has also joined us, Director Science City. So welcome. And now I request Dr. Marwaha to give inaugural remarks. Sir, please.
So, ma'am, thank you very much for your invitation to me to join this very important day. Well, I think we are here together on this occasion on the birthday of such a personality, actually, who is the base, who prepared the base of the modern biotechnology. Well, we are talking about the genetic code. I think he was the person who has started the work on that in 1968, how that is important in the cell and controlling the activities of the cells and controlling the proteins and all. Well, I'm really grateful to you for making me part of this program. Well, I am just very happy to see Felix also, because I had a small interaction with him during my stay at Bathinda. Well, I think he would make my job much more easier. The things which I would leave, he would pick up those things and explain it to the students and all the participants of this particular program and all. Well, talking about the biotechnology, well, ma'am, you talk about my role in the Knowledge City. Well, I joined the Punjab State Council Science and Technology in 1970 and 1997. Well, at that time, I was working as a professor and chairman, Department of Biotechnology at Punjabi University, Patiala. Well, when I was planning to join the state council as a director of biotechnology, people had been asking me this question, that why you are so young, you can be in any position at the university, why you are thinking of going to the council, joining as a director of biotechnology and all that. My answer was very simple at that time. I think I had been telling them while staying at Punjabi University, I know people up to Raipura, or people up to Raipura, they know me also. Let me shift to Chandigarh. At least I would be able to know people at Delhi, and Delhi people would also know about me. Well, when I came with some idea actually. Something was in my mind, and on that we started working on on the proposal of getting a center of excellence for biotechnology. Well, from 97 to 2004, till the Science Congress was held at Chandigarh. Well, sometime our proposal was approved by Government of India, but there were no share by the Government of Punjab, and sometime approved by the Punjab government, and no contribution from the Government of India. I think if everybody recall back, Professor Swaminathan actually came to attend that conference. Well, ma'am, if you remember, we invited him to Punjab State Council Science and Technology. Well, I just put a word to him. Rajan Kashyap was our Principal Secretary Science and Technology. I requested to him that sir, I want to ask a question from Professor Swaminathan. He said, Dr. Maroha, when you ask a question, be polite about, because Swaminathan is a big shot in biotechnology and the Indian administration also. Well, sir, then I told him that, sir, I would ask a very simple question from him, because I'm facing that question at the national level and all that. Well, I asked him a question which I was asked by the Government of India, that well, we are ready to fund this institution, but what sort of interaction it would have with the other national institution in the state? Well, the second question used to be that what would be the share of the state government? Then I told them that state is ready and state can answer that question, but those bodies where I was going to get the funds, I told them I'm not able to answer that, because I have no answer for that. I asked the same question from Professor Swaminathan. I asked that question from him in a different way. I said, sir, can you tell me, after independence, how many national institutions have come to the state of Punjab? He said, none. I said, sir, give us one, then see what we can do. And that was the turning point. And as a result of that, I think we got the Knowledge City, 3 national institutions in that, and the Center of Excellence of Biotechnology turned into a National Institute of Biotechnology and all. Well, I think from here, my journey as an administrator of biotechnology started. Well, I would share with all the participants. Well, as far as the definition, bookish definition of biotechnology is concerned, for me also it is very difficult. For most of you also would be very difficult to understand. But I simplify when I share this with my students also, or with everybody, or with the farmers of the state and the adjoining areas. I always tell them that useful application, technological application of the biological system is biotechnology. I think if you take it like that, you can understand the concept of biotechnology. Well, I think today's discussion on biotechnology, presentations on biotechnology is very important because of the pandemic of COVID-19, which we are facing, which is also a result of biotechnology. And we are trying to find out the solution also through biotechnology for developing the vaccines and all that. Well, as regard the applications of the biotechnology for the students, I think, and the areas of research of biotechnology which are of interest, it can be of interest to them. Well, I look at uh, 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 any science and technology subject and its application from 2, 3 angles. What is the strength of area where I am present, and what are the requirements of that area? I think we all know that agriculture is our strength. Based on that, we got to the Government of India for National Agri-Biotechnology Institute, which we have with us. Well, then second area I always say, because that is very difficult to ignore. Well, if you have something to eat, sometimes you eat in excess. With that, I think you fall sick. You need the support of pharmaceutical sector also. Very, very important. Well, it would be very, very difficult to segregate agriculture and pharmaceuticals. Well, with the taking over the chairman of the Punjab Pollution Control Board, my role has come into the management and prevention of the pollution. Well, when I look at my role and the role of biotechnology into this particular area, well, I find there are a number of issues which need attention, and for some of the issues, I think biotechnology can certainly help to settle those issues. Well, I just take 1 or 2 examples. Well, bioindicators and sensors, biosensors, well, is an area which is of much more importance and significance for any country which is into the controlling and managing the environment and all that. Well, with these sensors, we can sense out that at a particular stage or at a particular place, the environment is polluted because of a particular product and all that. I think this is an potential area by which one can control and at least assess the level of the pollution in a particular area and all that. And secondly, we are looking for some way outs to manage the pollutants. Well, I just take example of one air pollution being caused by the burning of the paddy straw. Well, there we are talking about uh, ex situ management, in situ management. Ex situ, we have number of methods, but with ex situ management of the paddy straw, we cannot take much of the paddy straw utilization because there are limited applications. The product which we produce by carrying out the ex situ management, I think certainly we have to have the market for those products. Well, if we don't have the markets, certainly then that would affect the in situ management of the paddy straw. Well, to me, the maximum application which find good for management of the paddy straw is the in situ. well, mulching the paddy straw into the soil and making it a part of the soil. Well, two, three things I would just like to share with you. This year, if you are going through the newspapers at the national level, I think two, three, or four different type of microbial consortia are being tried for decomposing the paddy straw. But the point is the gap between paddy harvesting and the sowing of the wheat crop is very, very short. The time duration is very short, window is very small. And the farmer is looking for an answer, want to have some way out by which it can decompose the paddy residues, paddy straw into the soil within that particular window. Well, different consortia are available. Punjab Pollution Control Board also has tried a consortia which is sold under the trademark of Relife, which is in the liquid form. Well, the consortia which we are using for that, well, if we are using it continuously, regularly for 3 years, I think at the end of the third year, that microbial consortia would be able to decompose the paddy straw, the mulch paddy straw, into the soil within 15 days that in that particular window. But first year it would take 30 days, and second year the time would, because the microbial consortia would develop, become a part of the soil, and then automatically it would start decomposing the mulch paddy straw and all that. Well, here the biotechnology can play a very, very important role. Well, when I say microbial consortia, is a combination of microorganisms. Well, the microbial consortia which are available in the market, they usually contain 6 to 11, 12 microorganisms as a part of that consortia. Well, they multiply in the soil as per their requirement and carry out the decomposition of the paddy straw. Well, here the biotechnology can play a very, very important role. Well, we have to identify that which of those organisms is required in the maximum number, and we have to increase their population, and that would certainly help to reduce the time duration for the decomposition of them. Now, what is the advantages of decomposing the mulch paddy straw into the soil? Well, the advantage is we are adding carbon and nitrogen. Decomposing it, we are adding carbon and nitrogen into the soil. Well, if you see, during the last 60 years, well, we have converted the soil of Punjab into sand. The water holding capacity of that soil has also reduced. You irrigate it, and as much as the requisite water is taken by the crop, and rest of the water just gets into the subsoil water and all that, or it is lost and all that. Well, by decomposing, you are adding carbon and nitrogen, which has the water holding capacity. You are increasing their concentration that has the water holding capacity. Well, the PAU research has established that at the end of the third year, well, the water requirement for the next water irrigation requirement for the next crop, that is wheat, at the end of the third year would be reduced by 20 to 25%. Well, this is one example I just wanted to share with you. Second, very important example. Well, I'm talking to the students actually. Well, when you go to the market, your requirement of the food have totally changed. Well, you are well, what we had been eating most of the time, chapati and rice. But when you are into the market, you are into the different type of foods. Well, processing of that. In some of them, the biotechnology plays very, very important role. Secondly, you are much more health conscious than we people. Than we people. Well, when you eat anything, you calculate the calories of the energies you have taken by eating that particular. You take a limited amount and calculated amount of the calories, and for that you have to have a specific food and all that. Biotechnology can play a very important role and is playing a very, very important role. Well, I had been to Canada in 2004. Well, I visited Quebec. There is a town known as Saint Hyacinth in Quebec. There is an Indian actually, Dr. Arora is there, Jatinder Arora is there. Well, he has done his BTech from NDRI Karnal, then his MTech as well as PhD. He did from in dairy technology with a specialization in biotechnology from University of Saskatchewan. Then he started with a pharmaceutical company, but he shared with me that in 97 or 98, he thought of that he has not come to the country, to Canada for that job. He said, I went to the counselor of St. Hyacinth that I just want to set up an industry which produces well high value milk product, which is called yogurt. Well, actually, 98 he started that company. 2004 we were there. He was preparing 7, 8 different types of yogurts and all nutraceutical products, and his turnover in 6 years was $150 million, Canadian dollars. I think the time is changing. You are changing. Well, there are a number of good areas where you can work. Third, I just would like to share with you the pharmaceutical diagnostics are very, very important. I think biotechnology is playing a very, very vital role in the development of diagnosis. I'm talking about a few examples which are very lively examples. Well, now the concept of diagnosing a disease has changed. Earlier, when we used to go to the doctor, they used to take blood and carry out the analysis and tell us which particular disease we are suffering from. Now some diagnostic kits are available, which are the result of biotechnology. With that, immediately they tell us that from what we are suffering, what is the level of our suffering and all. Well, similarly, vaccines are another things. I think for the younger generation, there are number of areas relevant to the strength of particular state or area are available. They can take up these research areas and can develop their careers. Well, I wish you all success to the younger generation. And before I conclude, I would like to say that whatsoever I have left, most of the things I think I have left, I think they would be taken care by Dr. Felix as well as Dr. Vandana. Thank you very much. Thank you.
You are muted, Kamaljeet.
Yes, ma'am, I was. Thanks a lot, sir, for sharing your views with us and with the audience as well. And now, before I request Dr. Felix to start his presentation, I take this opportunity to introduce him. Uh, Dr. Felix is an award-winning science communicator and YouTuber. Dr. Felix is based at Department of Botany, Central University of Punjab, where he is heading the department. Earlier, he was part of Indian Antarctic Mission and elected National Core Committee representative of Biology and Medicine at INIAS, a young science academy in India. Dr. Felix founded Young Academy of India, India's first young academy open for all disciplines of rational knowledge. He is also an avid cyclist, advocate for zero waste movement, and a complete organ donor. So this was a brief introduction about Dr. Felix. Over to you, Dr. Felix.
Yeah, thanks a lot, Ms. Karanjit Kaur, for introducing me. And what a surprise to see Professor Marwaha here, my former colleague. And I'm really happy to see you, sir. And yes, your whatever the words that you spoke is absolutely relevant on today, and especially on the paddy stubble. You know, the how to actually process the stubble on a sustainable way. You know, decomposition of stubble and everything. Whatever he said is really really profound. And yes, so I totally agree with Dr. Neelima's comment that Professor Marwaha is a very fertile person who brings a lot of fund. And here in the University of Central University of Punjab as well, that it was through the Professor Marwaha's efforts that we got a new department of applied agriculture. And he has brought us crores of rupees of the grant. And we have bought so many equipments. So thanks a lot, Professor Marwaha, for your a brief stint here in the university, but it was really productive. And my applause for you on your continued effort in the Punjab Pollution Control Board. And today my talk is, I can see that most of the participants are from the school. So, my talk is targeted towards the school children. And screen, let me share the screen. Yes, so this is Har Gobind Khorana's. Yeah,
Dr.
this
Felix,
I hope you
a
can see the
moment.
screen.
Dr.
Yeah.
Felix, just a moment. Could you kindly use English, because some of our students might not be very comfortable in English. So
I will try out. Yes. So yeah.
yes, if you explain complex concepts to them, the idea is that we explain all the complex concepts.
Right. So yeah,
you
I will
use a mixed language, whichever way is comfortable to you. Thank you.
Fine. I will give a try, because Hindi is also I'm not really too very good with Hindi. So yeah, I will give a try. I will make it very very simple.
Thank
Because
you.
this students are mostly the school students I can see. So let me share my screen once again. I hope you can see my screen. Am I right?
Yes, sir, it's visible to us.
Perfect. So yes, today's talk is entitled "How Har Gobind Khorana's pioneer works paved way for the development of COVID-19 vaccine." And this talk, I can also see among the participants, many are my own students. I can see Mohammed Singh. Hi, Mohammed Singh. Many of my own students have participated in this talk. Yeah, I'm really happy to see you all. So basically, this talk is about the work of Professor Khorana and how it led to the development of COVID-19 vaccine, especially mRNA vaccine, and also the recombinant adenovector vaccine. So what is the connection with the Khorana's work and the vaccine development? Because right now it is pandemic, right? COVID-19 pandemic. And right now in media, the talk is all about the vaccine. And not many people really understand that Har Gobind Khorana's pioneer work contributed in the development of the vaccines. So before that, let me take you to London. UK I've been twice. And this is a very famous museum in UK. It's called the British Museum. If you ever get a chance, please go to London. All the museums are free, completely free. You don't have to pay a single money in London. And more than that, photographs are allowed. There is no problem with taking the photographs anywhere abroad, you see, not just here in India. Of course, most of the museums, it's not allowed, but in abroad, everything is allowed. So the British Museum in London, if you go check out, there is a very interesting exhibit in British Museum. The exhibit is called Rosetta Stone. This is a stone, a पत्थर है, पत्थर के ऊपर लिखता है तीन लैंग्वेज में है इसके अंदर ऊपर क्या क्या लैंग्वेज है ऊपर वाला लैंग्वेज का नाम है हायरोग्लिफिक आपको मालूम है क्या है हायरोग्लिफिक बहुत पुराना वाला इजिप्ट का लैंग्वेज है हायरोग्लिफिक उसका नीचे है डिमोटिक देन ग्रीक so तीन language wala this stone is the most important piece of archaeological history because just because of the discovery of this stone now we know how to read and understand hieroglyphic the ancient Egyptian, the scripts. Ancient Egyptians had a great civilization, and they have written so many facets of their information on the hieroglyphic. So the person who decoded the hieroglyphs through the help of Rosetta Stone is Jean-François Champollion. He's a French linguist, and his work paved way for the entire field of Egyptology. And just like that, the work of Har Gobind Khorana is very similar. It is basically, as this is DNA molecule, right? So the DNA molecule have got 4 bases, that is A, T, G, and C. We have all learned in the school. And right now you're learning, right? The students are learning these alphabets and why it is very important. Now, these, by the way, who discovered this DNA double helix? Watson and Crick. So the Crick is a co-discoverer of the DNA helix or DNA molecule. And today morning also I had a talk, and in which I said a quote from the Crick. The Crick's very famous quote is that the dangerous man is the one who has only one idea, because he will fight and die for it. This is what happening in the world of terrorism, right? If you have only one idea, you are going to fight and die for it. That is what the dangerous man. Be open, be a student, friends. This is my #1 recommendation to all the viewers of today's program. Just forgo validation and ego. Just stay a student, a humble student, lifelong. I'm also a student now. And this is the DNA molecule. Now, if I give you a small stretch of DNA, CCTA, how do you read it? That is a problem, right? How do you encode this DNA? How to make sense of the random stretch of DNA? So double helix was discovered in late 1950s, but it was through the work of Khorana that we could now able to understand what these letters mean and how much is the word length of these DNA molecules. So there are only 4 letters. We all know that Watson and Crick have already revealed that the DNA molecule has got only 4 letters. Not 26 letters. Like in English, we have 26 letters, right? A to Z. But DNA has got just 4 letters: A, T, G, and C. But how to make words? Are words of different length, or words have the standard length? Yes, it's a standard length. And what is the length of each word? All these things remained unknown before the work of the Khorana. It was Dr. Har Gobind Khorana's work led to something called codon, triplet codon. So basically, the DNA molecule has got ATGC, and these are AUGC for the RNA, where the Har Gobind Khorana worked more on RNA rather than DNA. Both are nucleic acids. Now, these are coded in 3-letter words, and these words are called codon. So codon is nothing but words of our genome or DNA molecule. So every word has got just 3 letters, like cat, C-A-T, cat. It's an English word. That is cat is also 3 letters, right? Just like that, cat itself is a DNA molecule too. C-A-T. So just like that. So it's just 3-letter words. This and each word, there are, if you look for with 3 letters, and if you look at the combination, there are 64 different kinds of letters, right? And these letters code for different amino acids. And amino acids, these are nothing but building blocks of protein, isn't it? So there are 20 or 22 essential amino acids, and these 64 codons code for either amino acid or for start and stop codon. So this work, the genetic code work, is the main work of Har Gobind Khorana. So this led to our real deep understanding of the genome, how the genome works and what this triplet codon code for, which amino acid this is coding for. So all this is done by Har Gobind Khorana's pioneer work. Now, if you look at this, is a very recent paper I clipped from the Nature. So this is a genome of the SARS-CoV-2, that is severe acute respiratory syndrome, coronavirus serovariant 2. So that is what coronavirus, the novel coronavirus. So if you look at the coronavirus genome, it has approximately 29K. That means 29,674 letters, and of course each letter is divided into, 3 letters together forms codon, and it code for various proteins, right? You can see that various open reading frames. Don't worry much about this one, but these are all coding for protease, is an enzyme, papain protease. All these are various, enzymes and proteins that this genome of coronavirus code for. All this is possible because of the pioneer works of Har Gobind Khorana. So, if we don't know how to make sense of these codons, then we will never come to know about which section of its genome code for which protein. अभी हमको पता हो गया, देखो यहाँ A वाला ग्रीन वाला सेक्शन है, स्पाइक S लिख दिया इधर. So A वाला सेक्शन एक्सप्रेस करके बाद स्पाइक प्रोटीन S प्रोटीन सिंथेसाइज कर रहा है. This is in the coronavirus, on the surface protein, spike protein is being synthesized by the small region here, you see. So this is how we are actually knowing it. Now, coming to the vaccine, this is an HIV vaccine image, but of course the same thing for any kind of vaccine. So several kinds of vaccines. The oldest technology is whole inactivated vaccine. So basically you take the virus and you kill it or attenuate it and use that antigen to our body. That is called a very old technology. It is the first technology that we started developing. That is what Edward Jenner's technology, right? That is what whole inactivated. And that is a technology that Russians used against COVID-19. It's called Sputnik 5 vaccine. And one of the Indian company, Bharat Biotech, is also using Covaxin. That is also the same technology, very old technology, whole inactivated. But latest technology is RNA vaccines or recombinant DNA vector vaccines, So we have got this RNA vaccine or DNA vaccine. 2 companies are making very famous COVID-19 vaccine. One is called Pfizer BioNTech and another one is called Moderna. They both are using RNA vaccine, rather mRNA vaccine. And another vaccine by Oxford University is actually recombinant viral vector vaccine, not bacterial vector, but it's a viral vector vaccine. The virus is adenovirus, in which they express the spike protein. So both of these, all the 3 vaccines directly linked with the pioneer works of the Har Gobind Khorana. So because what he did it, now we can synthesize these vaccines. Oxford vaccine is also a generic version of the Oxford vaccine, the AstraZeneca's vaccine. Here in India, the Serum Institute in Pune is making it, so the Covishield vaccine, right? And the Covaxin is the old style whole inactivated vaccine. So let me first say about the story of Moderna. Moderna and Pfizer are quite similar, the mRNA vaccine. So how this vaccine work and how they develop this vaccine. American drug discovery company based in Cambridge, Massachusetts is what the Moderna is about. So by the way, what is drug discovery? Drug discovery means you are discovering a new drug or vaccine, right? So that drug discovery is very important for COVID-19, isn't it? So if you look at the timeline, last year, January 11, Chinese authorities sequence a whole genome and submit the sequence to NCBI GenBank. Much appreciations. If the Chinese are not that transparent, they will never reveal the whole sequence to the GenBank. GenBank is open. It's an open knowledge. Anybody can submit their sequence to the GenBank. Completely open access. And but not every companies or every organizations put their sequence to the GenBank free of charge. Of course, they release free of charge. It was happened in January 11, 2020, last year. It's almost 1 year back, right? And look, January 13, Moderna identified the gene coding for the spike protein. ये देखो इसके ऊपर spike देखा ना, or red लाल वाला वो वाला spike protein. What is the importance? A spike protein enable the virus to attach to our mucosal membranes like nasal membrane or alveoli of the lungs. All this mucosal membrane, this is what the virus can attach to. So this spike protein they identified within 3 days, friends. Just 3 days it took. January 11 they released. January 13 National Institute of Health of the US government and Moderna, the company, they identified the gene. See, how do they identify a gene? Because of the Har Gobind Khorana's work, you see. They synthesize a gene, that means oligosynthesizer. Artificially, you can make genes. Are you aware of it? It's a new technology, isn't it? You might think that, wow, synthetic biology is taking new dimension. Yes, you can synthesize genes just by looking at the sequence data, you know. So that is what they did it. So it's like, you know, the Americans are sending some message to Indians for how to build a three-dimensional structure, and we are making the three-dimensional structure. So information is transported by internet. See, internet is also very important building block of this COVID-19 vaccine, because thanks to the internet, you know, from Beijing, the code went to the US and they downloaded it, and then they synthesized it after identifying the gene responsible for the spike protein. They synthesized this as mRNA 1273. That's it. They developed the vaccine in just 3 days, record time. Look at that, just 3 days. That is what. So they manufacture, bulk manufacturing took 25 days, and then they ship for approval for phase 1 human clinical trial. What is this phase 1 trial, by the way? Yes, every vaccine or drug needs multiple trials, you know, to make sure that this works. So the medicine, the modern medicine is called evidence-based medicine. What does that mean? Evidence-based, because yes, evidence matters. Without evidence, no drug or vaccine should be allowed in the market, you know. And whatever the trials which they are making should be released to the private or the public. Everybody should, it should be open, the knowledge should be open, you know. Very important. So this is what, you know, if you look at this, how did they really identify that this region code for the spike protein? It's very difficult, right? So that is where the homology search comes in picture, like BLAST. So other coronavirus, by the way, coronavirus is a family of the viruses. It's not just one virus, but it's a family. Like Hominidae is a family of human beings, right? Different animals, like chimpanzees, also a part of Hominidae. Like that, you know, this is a family of coronavirus, the coronavirus family, related members. This region code for the spike protein. Then definitely this region will also code for spike protein in our novel coronavirus. That is how they are identifying certain regions in the genome, and this is known as you know, genome encoding, right? Or annotation of the genome is done by this process. And they, after synthesizing, they incorporated the synthetic spike protein gene into a functional mRNA. This is an mRNA, you see, 5' untranslated region, coding region, and poly A tail. So in between this mRNA is just one stretch of an RNA molecule, inside which they synthesized that spike protein gene and added it. You know, that is what now we call it as RNA vaccine, friends. How do they synthesize? Look, this is a new synthesizer. Here in Bathinda, we have all facilities. We also have the gene, you know, the sequencer, DNA sequencer we have, but we don't have oligosynthesizer. It's really expensive, you know, machine. I'm not sure in Punjab anybody has this. It's a very famous, you know, model is H8 SC oligosynthesizer. So this machine can synthesize the DNA molecule. So we just have to tell them the sequence, and the machine will synthesize it. See, this is what the synthetic biology, the latest invention, isn't it? You might think that it's really a latest invention, but friends, this machine or this technology, the oligosynthesizing, was discovered by who? Guess what? Again, Khorana, yes, in his work in 1970s, and this is one of the reasons why he was awarded the Nobel Prize. You know, so of course this comes in later after the Nobel Prize in 1968, right, when he won the Nobel Prize. So his work has paved way for synthesize. So he is the first person in the world to synthesize nucleic acid, you know. So that is exactly what we are doing now for synthesizing mRNA vaccines against COVID-19, you know. So there is a direct connection with COVID-19 vaccine and Har Gobind Khorana's work in 1970s, you know. So once you synthesize this mRNA, that is basically the viral spike protein you synthesize in that machine and incorporate into a functional mRNA. Then you cover that with a lipozyme and then inject it. That is what the vaccine is. Both this vaccine is 0.3 ml. Either BioNTech Pfizer vaccine or Moderna's vaccine, both are 0.3 ml, but contains different, the concentration is a little bit different. And both are mRNA vaccine, so mechanism is identical, but only that the storage, there is a difference. I'll come to it in a short while. Or you can ask me some questions, anything you want to know about this COVID-19 vaccine. So after injecting, these lipozymes combine to our own cell, and then this RNA gets translated because these are mRNA. So many fake news says that these vaccines has got nucleic acid, it will incorporate with our human genome. These are all fake news, friends. Never believe in WhatsApp University or Facebook University. Never trust those stories. It doesn't happen that way. So this RNA, that is mRNA, gets translated by ribosomes, and once it gets translated, this ribosome makes copies of spike protein. So a spike protein हमारा cell के अंदर बन रहा हो, they're manufacturing these spike protein inside our human cell, right? So the cell is course, I don't know, in Malayalam, my language is Malayalam, by the way. I'm educated in a Malayalam medium school, not an English medium school, right? So in Malayalam, we call it as kosham. Kosham means cell. I don't know what is the Hindi equivalent of that, but anyway, it's our own human cell, and in which this RNA gets entered, and this RNA gets translated, right? So the translation is a process by which it, the amino acids and proteins get synthesized. So these proteins are nothing but spike protein, because that is what we incorporated in, right? Recombinant, right? So these spike proteins get expressed inside the cell, the cytosol, and when this cell die, what will happen? Look at that. The cell dies. Right? Then these small spike proteins get bind with helper T cell. The helper T cell identify it and binds this molecule, this this spike protein, which is it's an antigen, you see. And then helper T cell gives this one to the B cell, presents this antigen to the B cell. B cell is what we are making antibodies. So B cell start making these antibodies, and antibodies are everywhere in our blood plasma against real COVID-19 infection. So after getting this vaccine, your plasma has got lots of antibodies. Antibodies are like our defense machinery, the military people, right? They have the gun and they're ready in case some intruder attacks our country. Just like that, the antibodies are everywhere in our blood plasma, and antibodies are ready to fight. And they're ready to fight with the intruding the virus. So whenever the virus comes inside our body, this antibodies attacks it and it just kills. That is what really happened, right? So that is what the RNA vaccine technology is all about. So RNA vaccine is that first step is that you have to identify which molecule, which gene to express, and then incorporate into a functional RNA, and then through lipozyme you inject it, and the rest of the work is all inside. Automatically it will be happening, because that is how our immune system works. So it gets activated, antigen presenting cell present that to the T cell present to the B cell start manufacturing the immunoglobulin M and G, and that renders a protection from the future infections. So in case you get infected later at your stage, and you are protected, friends. So that is what the mRNA vaccine is all about. By the way, the two mRNA vaccine which I introduced here, one is Pfizer BioNTech, which is approved in 20 countries in the world. And the other vaccine is Moderna vaccine, that is approved in 2 countries of the world, that is Canada and the US. The other vaccine is much more common, Pfizer BioNTech. BioNTech, by the way, is developed by a Turkish immigrant worker, an Islamic worker in Germany. So they developed this immigrant worker, Turkish immigrant. His name is Ugur Sahin. He developed this vaccine, and this is one of the most famous vaccine for the COVID-19, the BioNTech Pfizer vaccine. Unfortunately, none of these two vaccines are available here in India. So we have now two new approvals. The first vaccine which got approved is Oxford University AstraZeneca vaccine, that is Chadox nCoV-19. So Chadox is, is basically adenovirus vector vaccine developed by Oxford University team. That is why it is called Chadox vaccine by Oxford University. And Covishield is a generic variant of the vaccine, which is manufactured here in India by, Serum Institute of India at Pune. It's generic. It's the same thing. The technology is developed in Oxford University, you see. So, which is a recombinant viral vector vaccine using adenovirus. That is what this AD stands with, right? And another vaccine which is available in India is going to avail. Right now it is not. Right, to be launched soon in India is Covaxin by Bharat Biotech. I told you this is old technology. Sputnik 5 is also the same, or polio vaccine is also the same, which is inactivated virus, like polio vaccine or Sputnik 5. It's inactivated whole virus. It is basically whole coronavirus. You kill it and you inject it. That is what this vaccine does, right? So these two vaccines are one, but even for the Chadox vaccine, that is our AstraZeneca Oxford University vaccine, Har Gobind Khorana's work paved way for the development of this vaccine as well. So I have a YouTube, the introduction, the Miss Kamaljeet Kaur said about my, my channel. Yes, I do have a channel, and I do have a monthly science show called Curiosity, in which I cover what really moved the science the last month and what are the opportunities for the students. So please tune in. Right, the Curiosity is my thing, and my name is Felix Bast. Okay, you can just Google my name, or you can search my name in the YouTube to go to my channel. So please have a look in the Curiosity, and thanks a lot for inviting me here today to spend this brief time with you all. And I'm ready to take up any of your questions. So I would really love to get at least two questions from you. Nanni is Malayalam, that means thank you.
Thank you, sir, for such insightful information. Though we have a question from students, ma'am, दो क्वेश्चंस हम लोग अभी ले सकते हैं क्योंकि सर की एक और कहीं और भी उनकी एंगेजमेंट्स हैं, he has to attend another meeting. So अगर हम लोग दो क्वेश्चंस पहले ले लें.
Yes, of course. And
Yeah.
Dr. Felix, thank you. It was really, really great. I learned a lot from your talk. Thank you.
Thank you. Thank you, Nilima. Dr. Nilima. Yes, I'm sorry because my Hindi is really bad, pathetic. Yeah, I have to improve my Hindi. Yes, definitely.
That is perfectly fine.
Yes, sir.
I should learn Punjabi also now. It's been 10 years since I'm here. Yes.
Yes. So you can take questions. There were children who were raising hands.
Yes, yes, ma'am. There's a question from Amandeep. She said that, sir, as you already told that Covaxin is prepared from whole inactivated adenovirus, do you think that there is any chance of activation of this virus in our body? Or if it happens by chance due to X, Y, Z reasons, then is there any effect of that virus on human body?
Okay, so we really don't know anything about the Bharat Biotech Covaxin, So one of the most reputed scientists in India is Gagandeep Kang. Probably you know it. Gagandeep Kang is the only Indian woman who ever became fellow of prestigious Royal Society of London. Before the independence or after independence, till date, she's the only one with an FRS, And Professor Gagandeep Kang said that, well, it is a very mystery how the vaccine, it's a real mystery. So we really don't have any idea about how that vaccine, or does it really work or not, because the phase 3 clinical trial is not out. So in general, there is a trend. I have covered this in my channel as well, and I have written extensively, including the Hindu, about the prevalence of fake news. When COVID-19 was, came out in picture in last year, there is a lot of fake news about that vaccine, what it does and what it does not, and we have formed a separate group. I'm also part of the group, Indian scientists responsible for COVID-19. part of the group. So fake news keep on coming, and now the vaccine is in the market, and another array of fake news is there. The fake news says that if you take this vaccine, you become infertile. That is one of the fake news. And one nurse in Alabama died because of vaccine. No, that is another fake news. If you open the WhatsApp, you can see lots of fake news about vaccine. My simple answer is that inactivated viral vector vaccine, viral vaccine, not vector vaccine. The adenovirus vaccine is a viral vector vaccine, which is recombinant, you see. I think your question mixed up adenoviral recombinant vaccine with inactivated vaccine. Inactivated vaccine is not recombinant. It's recombination is a genetic engineering, you need technology for it. But inactivation is a very old technology, just kill it. You take the coronavirus, you just heat it to kill it, and then administer that vaccine. That is what Bharat Biotech, which they claim that they achieved it. It has got emergency approval. It is not full approval. Indian government never approved that vaccine yet. So if that vaccine works, if the evidence is substantiated, if they have extraordinary evidence, so that is one of my favorite quotes by Carl Sagan. Extraordinary claims require extraordinary evidences. If you're claiming that you have a vaccine against COVID-19, it's your responsibility to show the world what are the extraordinary evidence you have. So my simple answer to that question is that yes, it is safe. There is no chance that inactivated vaccine can regain to infect you. No, that doesn't work like that. So, the government approvals are really strict, and Indian government also have got a great drug approval authority, and they're really, really strict and stringent. And if they approve a vaccine, that means that the vaccine works and it doesn't cause the disease, and it doesn't work that way. Yeah. So vaccine is absolutely safe, and please do not trust anything that you read through WhatsApp or Facebook. Lots of fake news is everywhere, friends. Please don't trust that.
Thank you.
Any other question?
Yes, sir. There's another question. I hope Amandeep has got the answer to his question. And another question is from S. Sethi. So can scientists prepare synthetic antibodies from recovered COVID patients?
Okay, so there is already the antibody administration is already there. So basically, there is called what is it called? Convalescent plasma therapy. Convalescent plasma therapy means once you recover from COVID-19, the plasma gets taken. That is basically the blood, and then they centrifuge it, right, to get the plasma. And why plasma? Because plasma is where the antibodies are. Take the plasma and inject into the patient who is really suffering the COVID-19. So that is called convalescent plasma technology. That is already in use for COVID-19. You see. By the way, I'm also a blood donor come plasma donor. Every year I make sure that I donate my blood. So that is also the youngsters, whoever is watching this. The program, please consider signing up with our own Ministry of Family Welfare. Government of India has got a card called Noto card. I have Noto card, and I'm a proud organ donor, and my whole body donor, post death, my whole body will go to a medical college. So coming back to your question, the question is about the plasma, right? So taking the antibody, so that is called convalescent plasma therapy. That is, whole plasma is being used as a therapeutic, against COVID-19. Now the question is that can we get this, the antibodies out, sequence it, and then use it as a vaccine, or recombinant vaccine from the plasma? No, I'm not really aware of any such development. Maybe that's a good point. I don't know about it. You have to search it out. But anyway, what I would expect is that these antibodies, IgM or IgG, in recovered patients, they will definitely have antigens. Antibodies will have epitopes targeted towards the virus, right? So that is exactly what these recombinant vaccines are all doing. So we are, as of now, all vaccines are targeting the same part, that is basically the spike protein. So maybe the recovered patients must be having antibodies with epitopes for other antigens of the COVID coronavirus, which I'm not aware of. Excellent question, by the way.
Thank you, sir. And और कोई बच्चा अगर कोई क्वेश्चन पूछना चाहता है तो बच्चे प्लीज हैंड रेज कीजिए, सो दैट हम लोग जान पाए कि आप क्वेश्चन पूछना चाहते हो सर से.
There is a question I can see from Shreyasi Thakur. She
Okay.
says why DNA has a double helical structure, one. The second is why DNA has ACGT only and not something else.
Okay. Why it has got a double helix alone? So, well, that answer itself is quite complicated. Simple answer would be that why it is a double helix is that it is basically a lot more stable. The molecule is a lot more stable in the double helical form rather than the single helical. I mean, it's, if it's a single single stranded molecule. So the double helix increases the stability. And why it is only four molecule, which is I'm not really sure why it is only four possible bases are there. Why not a fifth or sixth? What is there any mathematical heuristics play picture? I'm not sure. And why it's only four? Why not three? Why not five? I don't know that. It's a very interesting, curious question. Yeah, this is the beauty interacting with the kids, and they have very interesting questions, and definitely established scientists have to learn a lot from the children, school children, and their questions are a lot more curious questions. I would definitely say that, rather than the questions coming from a learned audience. The kind of questions that the students pose are really interesting.
So same question kal pucha gaya tha. There was another webinar yesterday, and somebody asked the same question, and I asked them to attend this webinar to get the
Okay.
answer. So
Oh
hopefully
yeah.
you got the answer.
Maybe Dr. Vandana can help. The next speaker is Vandana, right? So
Yes,
yeah,
yes, yes, sir.
Vandana can able to look it up, or you can answer this question. I'm not really sure why only four bases, why not five, or why not three. So yeah, but it's actually deeply conserved. Maybe chance plays a role, because ultimately, Darwin. If you look at the Darwin's theory of evolution, the entire biodiversity on Earth can be traced back to just the root, right? Luca, last universal common ancestor. So in that ancestor, we had four bases only, not five kinds of base, or three bases. And because of that ancestral state, every single descendant of the tree of life have got these four bases. By the way, this is the same thing if you look in a bacteria or virus or whatever, plants or animals or fungi, whatever you look, all have got only four types of nucleotide bases.
Yeah. Dr.
Yeah.
Felix,
Yes.
in my opinion, this is Vandana.
Ah, yes. Hi, Vandana. Yeah, tell me.
Hi. I think nature is making only those things which are actually required. And there is no requirement of fifth base, I think. All the proteins which are made up of 20 amino acids are fully coded by these four nucleotides. I think there is no need of fifth base. That's why nature didn't make it.
Yeah, well, that could be a possibility. Four can code it, but then why not three? What is the problem with the five then? So, and why only 20 are essential amino acid? So we could
Yeah,
have
that
had
is.
more essential amino acids too, right? That is
Yeah, yeah.
also a question here.
Yeah,
And
yeah.
which came first, protein or the DNA? Which came first? That is what many people now say, that it's RNA that came before the DNA.
Yeah,
Ribozyme, or
yeah.
RNA
Yes.
world, because RNAs can have both the functions together. Protein, I mean the enzymatic function as well as the coding function. So it's really an exciting question. Why not three or five? Why four? So what is so special about four? If you look back in culture and civilization or epics, or you can see that number three has a lot more significance than number four, right? Trilogy, for example, in Hinduism, for example, or Christianity. Number three has got a huge significance, but not number four. But maybe the nature works in that way, or maybe it's simple chance. Just that last universal common ancestor had four bases, and that is the reason that we now have got the four in all of the organic world.
So thanks a lot, Dr. Felix, for being with us today and for sharing your sheer knowledge with us. Thanks a lot.
Thank
And
you, ma'am.
moving further, I would request Dr. Vandana Jaiswal to start the presentation. But before, here's a brief introduction about her. Dr. Vandana Jaiswal is scientist, CSIR, IHBT Palampur, And her research areas are molecular biology and crop biotechnology, and in particular, marker assisted breeding. And he has been awarded with CSIR Young Scientist Award 2020 and CSIR Senior Research Fellowship, CSIR Nehru Science Doctoral Fellowship, and DST Women Scientist 2014-2016. So this was the brief introduction about her. Ma'am, please start with your presentation.
Thank you so much, Dr. Kamal, for nice introduction. Uh, it is a very good evening to one and all. It is my pleasure to be here and to be part of this event. And I must thanks Pushpa Gujral Science City for inviting me to interact with such a young audience. Now I'll share my slide. I think it is visible.
Yes, ma'am, it's visible.
Okay. So,
Ma'am, please begin with the slideshow so that it will.
now it is okay?
Yes.
Okay. Today I will be talking on the molecular biology journey, starting from the DNA to the genome synthesis. My colleague, my co-speaker, Dr. Butts, already told very much about the Nobel laureate Professor Khorana, and our our DG Pushpalata Science City already told about the bibliography of the Professor Khurana. He was born in the Raipur in village Multan. It is now in Pakistan. He was an Indian American biochemist. He got Nobel Prize in 1968 in physiology and medicine, and he shared his this Nobel Prize with Dr. Nirenberg and Dr. Hawley. Apart from this particular Nobel Prize, he was awarded with several other very distinguished awards like Gardner Foundation International Award, Lucia Gross Hortis Prize, for Women's RS, Albert Lasker Award, Padma Vibhushan, Willard Gibbs Award. Khorana also was the first person who chemically synthesized the oligonucleotide. Dr. Butts already told you about this. Since this particular webinar is for the students of class 10th and 12th, so I thought I should start with some basics of the molecular biology, which you already know. Maybe you are knowing it, but I thought first I should give this particular slide. We all know the cell is the functional unit of the life. हम सबको पता है, आपने पढ़ा भी होगा. सेल के अंदर एक न्यूक्लियस होता है, and nucleus is containing the genetic material which is called as DNA, and we know this DNA is the actually part which is transferring from one generation to the next generation. हम सब बोलते हैं ये बेटा है, इसके कैरेक्टर इसके फादर से ऐसे मिलते हैं या मदर से ऐसे मिलते हैं. तो एक्चुअली वो क्या चीज़ है जो कि हमारे जो कैरेक्टर्स होते हैं वो उनको ट्रांसफर करता है फ्रॉम पेरेंट टू द चिल्ड्रन. This is actually DNA, and DNA is the genetic material, and it is actually controlling all the features of any organism. And this DNA is made up of, you know, sugar base and 4 nucleotides A, T, G, and C. This particular DNA is transcribing to develop messenger RNA, and this messenger RNA then transform into the protein, undergo the translation process and make some proteins. Then the question arises how the genetic information coded here in a messenger RNA transfer to the protein. How amino acid is getting to know which amino acid is going to bind or which amino acid is not going to bind. Here, Dr. Khurana's work paved a vital road. Actually, on messenger RNA, codons are there. Genetic information written in the form of nucleotides, that is GATGNC. And Dr. Khorana said that there are 3 nucleotides which are making codons. Since there are 4 nucleotides, there will be 64 genetic codons, and they are encoding 20 amino acids. For now, we know the proteins, all proteins which are occurring in any organisms are made up of 20 essential amino acids. So these 64 genetic codons is actually encoding the 20 amino acids. And all of these codons, there are certain codons which are actually initializing the protein synthesis, which is AUG, and it is encode the methionine. And there are some stop codons which do not encode any amino acid, thus protein synthesis will stop. These are the checkerboard where different codons are written and which encode the different amino acids. अब मैं आपको थोड़ा इंट्रोड्यूस करना चाह रही हूँ कि हाउ दे आर नोइंग कि which codon is actually responsible for which amino, what are the experiments they performed. First, they have taken self-extract. Self-extract means they are containing all the machineries which are actually required for the protein synthesis, but they do not contain the DNA or RNA or amino acids. Means machineries are available, but raw material is not available. This is called a self-extract. In this self-extract, they are included synthetic mRNA, means they know what is the sequence in this particular oligonucleotide. For example, this is the poly-U. And then they added some radioactive labeled amino acids. And after sedimentation, what they found, since here only U is there, means only uracil is there, they got only phenylalanine in the precipitation. इसका मतलब क्या हुआ कि अगर poly-U होगा तो सिर्फ phenylalanine ही मिला. इसका मतलब poly-U is actually responsible to encode the phenylalanine, right? Now, in the same way, जब उन्होंने synthetic poly-C डाला, then they found only prolamine. जब उन्होंने सिर्फ poly-A डाला, then they find only lysine. It means this is how they identified which code is responsible for which amino acid, right? This is how entire encoding of 20 amino acids could be identified. Okay. Now, this is okay कि messenger RNA पे आपको पता है कि ये कोड है and this is the amino acid, but this is the nucleotide and these are the amino acid. They cannot bind to the amino acid. Then how these amino acids are present in the cytoplasm, and this is written on the messenger RNA. How these amino acids know कि अब हमें वहां जाना है और हमें वहां attach होना है? Who is actually transferring amino acid to the mRNA? This is transfer RNA. Transfer RNA is actually RNA, and it is clover leaf shaped structure. There are 3 loops: D loop, T loop, and anticodon loop. And here is the tail. This tail is actually contain amino acid, and this is the anticodon. It is the complementary region of the mRNA. If AUG is there, here will be anticodon, which is complementary to AUG, and here will be the methionine. So once messenger RNA transcribed from the DNA and came into the cytoplasm, transfer RNA will go to it and complementary transfer RNA will bind to it, and then corresponding amino acid will be binding. And this is how protein synthesis is going on. Once this protein synthesis is reaching to the a stop codon, here it is UAA, protein synthesis will be stopped. This is how a protein will be synthesized, starting from the DNA, then messenger RNA, then transfer RNA, and then finally protein. Now, अब ये है कि we know the how DNA encoding the proteins. But what is the utilization and application of this molecular knowledge in the societal impact? हमें पता चल गया कि DNA होता है. This is all about the knowledge. But how it can be applied in the social benefits? So today I am going to talk on the two topics which are most advanced, we can say, molecular techniques. One is genome editing and other is synthetic genomics. Genome editing allow us to modify the genome or modify the gene. With the help of genome editing, we can add, we can remove, or we can replace a particular segment in the gene. So once we know which is the gene, what is the sequence, what is the desirable sequence, what is the undesirable sequence, then we can actually correct the genome. This is called as genome editing. In genome editing, most precious thing is the enzyme, which is actually cut the DNA from a particular site, which is called as nucleases. And it is most crucial part because specificity of cutting a genome, a particular site, is very much important. There are several genome editing tools are available. It was started in 1919 until 2013. There are 4 different genome editing tools are available: meganucleases, zinc finger nucleases, talents, and CRISPR. CRISPR is most advanced technology, and this technology is being utilized nowadays. These technologies are mostly different in terms of their specificity. With the help of CRISPR-Cas, you can edit a single base. Suppose you know this is A. If A is there, this will be the amino acid and this will be the protein. And this protein is not functional. But if it will be a T, then protein will be functional. Then we can correct a single base within the genome. So this is the beauty of this particular tool. How this works? Suppose you are having this particular DNA. You know this particular part, this red part is undesirable, and we want to remove it, and we want to insert this particular yellow part in that gene or genome. So CRISPR-Cas, first we cut it, replace it, and then insert with the help of tools. CRISPR-Cas is very much exciting technology, and it is most precise technology currently available, and it is relatively simple. Multiplex genome editing can be performed through the CRISPR-Cas, and most importantly, it is very much cost effective, means cheaper, and very much easy to do. Now we can edit the genome. Now I'll give you some examples where, since I am basically a plant biotechnologist, so I will give you some examples where this CRISPR-Cas technology is utilizing to improve the crop, either production or any other traits. हम सबको पता है कि जो हमारी ग्लोबल पॉपुलेशन है वो डे बाय डे बहुत ज़्यादा इंक्रीज कर रही है, एंड देयर इज अ very big challenge before the breeders to feed such huge population, and not only the feed, but food should also be nutritionally rich, so that it will be to ensure the food security as well as nutritional security. What breeders do? Breeders basically what they are doing, they are making crosses and then transferring the characters from different plants and make some new varieties. They are developing new varieties, but development of a new variety is a long-term process. It may sometime takes more than 10 years. Second approach is genetic engineering. In genetic engineering, we are transferring some genes which are present in any other species to improve some plants. For example, if we talk about the BT cotton, we all know about the BT cotton. In cotton plant, there is a huge problem of stem borers. So what scientists do, they identified one gene, cry1ac from Bacillus thuringiensis, and transferred into the cotton plant. And now the BT cotton plants are basically resistant to the bollworm. So, but in this approach, there is a big problem of the biosafety issues. So in India, GMOs are not allowed. So third approach is genome editing, which is based on the CRISPR. In genome editing, we just correct a gene for development of new variety. Now this is the wet lab experiment. If you want to genetic editing in the plant, what you have to do? First, you have to construct a develop gene, and then insert through the different approaches are available to insert into the plant, then transformation of callus, then plant will be regenerated. And through the screening and selection of these plants, you can select plant where you found the inserted genes of your desired characters, then phenotypic screening and development of the variety. This is the most recent example of the application of CRISPR-Cas technology for the development of the golden rice. You know, rice में beta carotene नहीं होता है, but beta carotene is very much important and nutritionally it is very important. So using this particular technology, scientists are able to develop golden rice where they have edited one gene, and in that plant beta carotene is synthesizing in the grain, and grain colors become yellow. Second example is from tomato. हमें सबको पता है कि प्लांट में डिजीज की बहुत प्रॉब्लम होती है, और डिजीज से क्या होता है, यील्ड भी अफेक्ट होती है और क्वालिटी भी अफेक्ट होती है. देन एक जीन है SL-MOL1, सोलेनम लाइकोपर्सिकम MOL1. इसमें जीन में जब ये वाइल्ड टाइप जीन था, इसमें ससेप्टिबिलिटी बहुत ज़्यादा थी, मीन्स डिजीज बहुत ज़्यादा होती थी. जब इन्होंने CRISPR-Cas के थ्रू इस जीन में एडिटिंग की, देन व्हाट दे फाउंड रेजिस्टेंट प्लांट्स. मीन्स अगर आप इसमें पॉल्ट्री मिल्ड्यू का पैथोजन आता भी है, तो इस प्लांट को के अंदर रेजिस्टेंस डेवलप हो चुकी है, just due to the modification in one gene. This is another example from rice. In rice, there is problem of bacterial blight disease. By editing of one gene, scientists could be able to develop the resistant plants. Here, just editing in one gene, tomato plants were showing branches, and these branchings directly affect the yield. Yield were gone to double, or we can say more than double. So this is how genome editing technology is helping the breeders to develop new varieties with improved characters, improved resistance, improved yield. These are several examples from different plants, including bread wheat, cucumber, Arabidopsis, rice, tomato, potato, cassava, where genome editing technology is being utilized to develop improved plants. Now coming to some human example, a genetic disease called as Leber's congenital oromasis. It is in this disease what happened, the retinal cell get degenerated, and due to this degeneration, a blindness occur in childhood stage. It is a genetic disorder and there is no treatment available for this disease. In last year in Nature, this news comes that scientists identified there are some mutation in particular gene, and scientists are trying to edit this particular mutation in the gene so that this degeneration of retinal cell could be stopped and there will be no blindness. This particular research is under clinical trial. CRISPR-Cas not only the improving the plant system or we can say gene editing, but CRISPR technology is also utilizing for the testing of novel coronavirus. हम सबको पता है कि अभी टेस्टिंग की सबसे ज़्यादा प्रॉब्लम थी कि कौन सा मेथड बहुत प्रिसाइज है, कई बार फॉल्स नेगेटिव आते हैं, कई बार फॉल्स पॉजिटिव आते हैं. तो CRISPR-Cas मेथड इज द मोस्ट रोबस्ट मेथड for the testing of coronavirus. And here what they used streptavidin capture RNP bound biotilated substrate. If virus will be there, you will find a stain in this particular band. And this particular kit was developed by Dr. Devi Jyoti Chakravarti and Shobik Maitri at CSR IGIB. Now coming to the synthetic genomics. In synthetic genomics, what happened? Scientists are chemically synthesizing the whole genome. In genome editing, they are just editing some genes, 1 gene or 2 genes. But in synthetic genomics, they are actually synthesizing the entire genome. And the synthetic biology is also, Dr. Khorana played a vital role in the synthetic genomics also, and he was the first person who synthesized the gene. Although it was not the protein coding gene, it was the gene which encode the transfer RNA, alanine. It was in the yeast, but after that, several protein coding genes has been chemically synthesized. This is the journey of synthetic biology or DNA biotechnology. In 1996, the concept of minimal genome put forward, and polio first chemically synthesized polio virus was done in 2002. And George Craig Venter Institute is actually a leading institute for the synthetic biology. It is in the USA. And they are actually leading in the area of this synthetic genomics. And so these are the some genomes which are chemically synthesized, or we can say artificially synthesized, and they are vital. Now, concept of minimal genome. We know in our in any genome there are some essential genes and there are some non-essential genes. That means there is no need of genes there. So the scientists thought, if we remove those genes from the genome, and if cell will able to survive, and ये और क्या हमें help करेगा कि which genes are actually essential for life and which genes are not essential for the life. So at JCVI, they first design and synthesize the minimum genome bacteria, that is Mycoplasma genitalium, and it is of 531 kilobase pair and having 473 genes. Although the original genome size of this Mycoplasma genitalium is around 1000 kilobase. So though they dropped the genome size about half, and even then this particular bacteria survived. Now coming to the recoding of genome, means हमें ये पता है कि मिनिमल जीनोम है और ये जीन एडिट कर दे तो ये हो जाएगा, बट क्या हम दोबारा से रिकोड कर सकते हैं जीन को? Just 1 year back in 2019, this development has been done. Chin and his colleague actually recoded the entire Escherichia coli genome, and he made changes in 18,000 codons. What they did, actually, if if you know, serine is an amino acid which is actually encoded by the 6 different codons. Then what they found, if some of these codons can be removed from the genome, and only they utilized only 4 codon to encode this particular amino acids. Similarly, they, out of 3 stop codons, they used only 2 stop codon to be there in the genome. And finally, they synthesize the bacterial genome, and this bacterial genome is the fully functional. All the genome I talked about was either virus or bacteria, but there was no eukaryotic genome. Synthetic synthesis of eukaryotic genome is still going on. It is in the yeast. Yeast is having 16 different chromosomes, and these are different institutes which are involved in synthesizing the 16 different chromosomes of the yeast. This is undergoing project, and hope we will get some good news to synthesizing the whole genome of the eukaryotic genome. So now to the conclusion, DNA word is of 3 letters, which is called as genetic code. It is genetic code is actually responsible for all the features of the organism. Molecular tools are continuously emerging. Advanced technologies like CRISPR-Cas is continuously being utilized for the crop improvement and human health. And synthetic genomics is the new era of molecular biology. And with the help of this particular technology, we can design a new genome with desired characters. Thank you so much.
थैंक यू डॉक्टर वंदना। बड़े सिंप्लीफाइड वे में आपने बच्चों को समझाने की कोशिश करी है। देयर आर क्वेश्चंस ऑलरेडी कमिंग, तो वो हम लोग ले लेते हैं। एस वांटेड टू आस्क क्वेश्चन। एस सिटी प्लीज अनम्यूट योरसेल्फ एंड आस्क योर क्वेश्चन। एस सिटी, यस बेटा पूछिए, अपना क्वेश्चन पूछिए।
Ma'am, am I audible?
Yes.
Ma'am, actually I want to ask, like suppose there is some new strain of bacteria or virus, then how scientists will know about how they synthesize primer for that new strain of bacteria or virus?
For the primer synthesis, first you have to know about the sequences. If it is new virus, you may use the comparative genomics approach. You can find some other member of which belonging to the same family, and using the comparative genomics approach, you can synthesize primer from there. But you have to validate in particular sample.
Okay, ma'am. Ma'am, I have one more query.
Sure.
Ma'am, like you told us about CRISPR-Cas9 technique. Ma'am, how CRISPR-Cas9 technique, how CRISPR-Cas9 identify the undesirable sequence for gene editing in any organism?
Actually, for CRISPR-Cas, you first need to design the construct, and the designing of this particular thing is very much important. CRISPR-Cas is not identifying the particular sequence. There is a nuclease which is called as Cas9. This nuclease is actually, we are actually adding some part in the form of oligonucleotide, which is complementary of our gene or our sequence, which we want to modify. Okay, so sequence is there in your construct, and on the based on the complementary, Cas9 could identify the specific place.
Ma'am, so first of all, we have to construct
design. Construct designing.
Yes.
Construct designing is very much important and very much crucial to to do the precise editing.
Okay, ma'am. Thank you, ma'am.
Thank you, beta. I hope you have got the answer of your question. And yes, any other questions? Please raise the hand. If any other, and if somebody wants to ask any question, just raise the hand. Yes, Muskan Preet, please unmute yourself, beta.
Good afternoon, ma'am.
Yes.
Ma'am, I have a question that you said that the genome editing is very important and playing very important role in plants also. So there was no any side effect on plants of genome editing.
No, this is the beauty of the genome editing. In transgenics development, there is some side effect in not in plants, because in genome editing, what we are doing, we are just editing some particular sequence. We are not adding up different things into the plant. We are not adding different things. We are just editing particular sequence to get the desired product. And what developed through the genome editing technology, the developed plant will be the as natural as other plants are. Okay, so वहां पे आप जो प्लांट डेवलप होकर के आते हैं ना, that is totally natural. You can say, if you know about the alleles in nature, for a particular gene, number of alleles are available. Okay, and this is why even in case of human, हम सबको पता है हम सारे humans हैं, but सबके features अलग है अलग, क्यों अलग है? Because genes are there, but genes, there are different alleles for each gene. So basically we are actually creating a new allele only, and we know by editing this particular sequence what will be the protein and what will be the feature. So I don't think there is any side effect to the plant.
Thanks a lot, ma'am. And Muskan, I hope you have got the answer to your question. So,
but I'll also suggest Muskan, for example, अगर हम कोई कन्वेंशनल ब्रीडिंग या कोई और ब्रीडिंग करते हैं, उसमें हम क्या करते हैं, क्रॉसिंग करते हैं, दो प्लांट के कैरेक्टर्स को मिक्स करते हैं. उसमें क्या होता है, कई बार हम डिजायर्ड कैरेक्टर्स को तो चाहते हैं, बट उसके साथ साथ बहुत सारे अनडिजायरेबल फ्रैगमेंट भी इंसर्ट हो जाता है, जिसकी वजह से कई बार क्या होते हैं, आपके कुछ फीचर्स जो है वो डाउन चले जाते हैं. इन दैट केस कुछ अनडिजायरेबिलिटी प्रोड्यूस हो सकती है. बट इन जीनोम एडिटिंग आई डोंट थिंक सो कि कोई अनडिजायरेबल या साइड इफेक्ट आएगा प्लांट पे.
okay, thank you, ma'am. Thank you, Dr. Vandana. And now I request Dr. Rajesh Grover, Director Science City, to propose vote of thanks to one and all.
Thank you, Kamal, and it's really an honor to have been asked to propose a vote of thanks at the occasion. And first of all, I would like to propose a hearty vote of thanks to our chief guest, Dr. ISS Nawab, Chairman, Punjab Pollution Control Board, who has just left, I think.
Yes.
Who was with us, and this afternoon he joined and graced the occasion. And I, on behalf of the entire team of PGSC and as well as on my behalf, I express my gratitude to him for sharing his words of wisdom and giving an overview of the development of biotechnology in the state of Punjab, as well as inspiring our young audience. And I must mention our deep sense of appreciation for our esteemed speakers, Dr. Felix, Associate Professor at Central University Bathinda, and Dr. Vandana Jaiswal, Scientist at IHBT Palampur, for giving an excellent coverage to Dr. Har Gobind Khorana's discovery of genetic code, as well as how it has opened new avenues to the modern biotechnology, and as well as understand how this molecular and genetic mechanism has understanding has resulted in the successful implementation of the several human health and crop improvement programs. And I would also like to express my gratitude to our worthy Director General, Dr. Nilima Jareth, for her presence and gracing the occasion. as well as inspiring words at the occasion. Finally, I take this opportunity to acknowledge the support received from my colleagues to organize this webinar. And I would like to extend my special thanks to Engineer Ritesh and Ms. Kamal for providing all sorts of logistic support in organizing this webinar and make it a success. Last but not the least, we are grateful to our enthusiastic participants for their patient listening and joining us on this online platform. Once again, thank you everybody.
Thanks a lot, sir. Thanks all for being with us for today's webinar. Keep joining us. Keep in touch with us for coming webinars. There will be another webinar on 12th as well on National Youth Day. Please do attend that webinar also. Thank you all.